food and water move across large distances in plants. However, in plants, the movement of food and water also takes place across short distances. That is, within a cell or from one cell to another. This movement of food and water across short distances takes place either passively by diffusion or by active transport. In diffusion, the molecules move in a random manner, generally moving from a region of high concentration to lower concentration. Diffusion is a passive process. It requires no expenditure of energy. It is a slow process, independent of a living system, and can take place in all states: liquid, solid, and gaseous. Did you know that diffusion is the only means of gaseous transport in plants? Gases such as oxygen and carbon dioxide enter and exit stomata via diffusion. The rate of diffusion is affected by several factors, such as the size of molecules, their solubility in lipids, and the concentration gradient. Of these three factors, the concentration gradient is a must for diffusion to even take place. As for size, the bigger the molecules, the slower the rate of diffusion. And since all membranes are made up of lipids, lipid-soluble substances diffuse faster. For this same reason, hydrophilic or water-soluble substances find it difficult to diffuse through membranes. In addition to their existing concentration gradient, diffusion of such hydrophilic substances has to be facilitated by special proteins called transport proteins. This type of diffusion, facilitated by proteins without any energy expenditure, is referred to as facilitated transport. The rate of transport in facilitated diffusion is maximum when all transport proteins are used or saturated. Facilitated diffusion has an advantage in that it allows cells to select substances for diffusion. It is, however, sensitive to inhibitor molecules that react with side chains of the transport proteins. Proteins are responsible for creating channels in the membrane, which allows molecules to pass through them. Some of these channels are large enough to allow a variety of molecules to cross over. Some of these channels are always open, while others are controlled. Some transport proteins, called porins, form pores in the outer membranes of cell organelles, such as plastids or mitochondria, to allow molecules of the size of small proteins to pass through. When a molecule binds itself to a porin, it rotates to release the molecules inside the cell. The water channels are made up of 8 different kinds of aquaporins. Some transport proteins allow diffusion if 2 different types of molecules are required to be transported. Based on direction, transport of molecules can be differentiated into 3 types: symport, antiport, and uniport. Both molecules cross the membrane in the same direction in a symport, while they move in opposite directions in an antiport. In uniport, as the name suggests, a single molecule diffuses across a membrane independent of other molecules. Unlike diffusion, active transport requires energy to move molecules against the concentration gradient. It is carried out by membrane proteins that use energy. These proteins, called pumps, can also transport substances from low concentration to high concentration, which is taken to uphill transport. Just like facilitated diffusion, the active transport rate is maximum when all protein transporters are being used or saturated. Also, these proteins are specific to the type of substances they carry across. They are sensitive to inhibitors that react with their protein side chains. Of all the means of transport discussed, both diffusion and facilitated diffusion always take place along a concentration gradient and without the expenditure of any energy, whereas only active transport uses energy. Plants need to be watered. However, have you ever wondered how seeds absorb water in the first place? Seeds absorb water by a special type of diffusion called imbibition, where solids absorb water, causing an increase in their volume. Imbibition is a special type of diffusion where, once again, water movement is along a concentration gradient. For any solid to imbibe a liquid, it must have an affinity for the liquid. Also, the water potential gradient between the absorbent, that is the solid, and the liquid imbibed is essential for imbibition. The water potential gradient comprises two main components, solute potential and pressure potential. To understand the concept of water potential, we must take into account that water molecules possess kinetic energy. They are in rapid and constant motion in both liquid and gaseous form. The greater the concentration of water, the greater its kinetic energy. As a result, it has greater water potential. Water potential is denoted by the Greek symbol psi. It is expressed in pressure units as Pascal or PA. The water potential of pure water at standard temperature and pressure is taken as 0. If a solute is dissolved in pure water, the solution has fewer free water molecules. This decreases its water concentration, resulting in decreased water potential. Therefore, we can easily conclude that pure water has the greatest possible water potential, whereas the water potential of all solutions is less than that of pure water. The magnitude of the lowering of water potential due to the addition of solutes is called solute potential. Solute potential is represented by the letter psi s, and it is always negative. Solute potential becomes increasingly negative with the addition of solutes. When two solutions come into contact, water molecules from the system with higher energy will move to the one with lower energy. That is, movement will take place from the solution with higher water potential to the one with lower water potential. For a solution at atmospheric pressure, its water potential is equal to its solute potential. But if pressure greater than atmospheric pressure is applied to pure water or a solution, it results in an increase in water potential. When water enters a plant cell due to diffusion, it builds up pressure against the cell wall, causing the cell to become turgid. This pressure that is applied by the protoplasm against the cell wall is called pressure potential. Pressure potential, which is denoted by psi p, is usually positive. However, negative pressure potential is critical to water transport up the stem via xylem cells. Hence, it can be stated that the water potential of a cell is a combination of its solute potential and pressure potential. A plant cell is bounded by a cell wall and a cell membrane. It also contains a large central vacuole. This vacuole contains a vacuolar sap that contributes to the solute potential of the plant cell. While the cell wall is freely permeable to water and other substances, allowing them free movement, the cell membrane, on the other hand, is a semi-permeable membrane. The cell membrane, together with the vacuolar membrane, which is also called tonoplast, determines the movement of molecules in and out of the plant cell. The diffusion of water across a semi-permeable membrane like the cell membrane is referred to as osmosis. Osmosis occurs spontaneously in response to a driving force. The net direction and rate of osmosis depend on two factors: concentration gradient and pressure gradient. If two solutions are put into a chamber separated by a semi-permeable membrane, then water will move from the region of higher chemical potential or concentration gradient to the region of lower chemical potential until equilibrium is reached. At equilibrium, both chambers will have the same water potential. Also, the potential of water molecules to move from a hypotonic solution to a hypertonic solution across a semi-permeable membrane is called osmotic potential. A hypotonic solution is a solution that has more water and less solutes, whereas a hypertonic solution is one that has less water and more solutes. Let us consider another example of osmosis. A solution of sucrose in water is put into a funnel and separated from the pure water in a beaker by a semi-permeable membrane. The water, being hypotonic, will move from the beaker to the funnel containing the hypertonic sugar solution, raising the solution level in the funnel. This water movement will continue till both solutions in the funnel and beaker achieve equilibrium. Now we can apply external pressure to the upper part of the funnel so that no water diffuses from beaker to funnel through the membrane. This external pressure, required to prevent the water from diffusing, is known as osmotic pressure. Osmotic pressure is a function of solute concentration. The higher the solute concentration, the more is the diffusion of water. Therefore, more osmotic pressure will be required to stop the entry of water molecules into the solution. The water potential is equal to its solute potential for a solution that is at atmospheric pressure. Unlike osmotic potential, which is negative, osmotic pressure is a positive pressure. However, numerically, osmotic pressure is equal to osmotic potential, only their signs differ. Osmosis, therefore, plays an important role in water movement in the plant cell. Movement of water in and out of a plant cell is essential to a plant's existence. The plant cell wall can either shrink or become turgid in response to the movement of water. It is the surrounding solution of the cell that decides the direction in which water flows. The surrounding solution can be categorized into three types: isotonic, hypotonic, and hypertonic. When the concentration of the external solution is the same as that of the cell cytoplasm, the solution is said to be isotonic. Such a solution balances the osmotic pressure of the cell cytoplasm. If the concentration of the external solution is lower than that of the cytoplasm, it is said to be hypotonic. On the other hand, if the external solution is more concentrated, that is, it has more solutes, it is said to be hypertonic. If a plant cell is placed in a hypertonic solution, water moves out of the cell due to osmosis. The cell membrane shrinks away from the cell wall with this outward movement of water. This phenomenon is called plasmolysis, while the cell is said to be plasmolyzed. In plasmolysis, water first moves out of the cytoplasm and then the vacuole. This movement of water takes place from a cell which has higher water potential to an area outside the cell that has lower water potential. However, plasmolysis is a reversible process. When plasmolyzed cells are placed in a hypotonic solution, that is, a solution with less solutes and higher water potential, water moves from the solution into the cell. This causes the cytoplasm to build a pressure against the cell wall. This pressure is called turgor pressure. This turgor pressure exerted by the protoplast against the cell wall due to the entry of water is called pressure potential Ψ-P. It is the turgor pressure in the plant cells that helps a plant to be erect. Since plant cells have a rigid cell wall, the cell does not rupture despite the turgor pressure. However, the cell enlarges and results in the extension growth. Now, if this plasmolyzed cell is placed in an isotonic solution instead of a hypertonic or hypotonic solution, there is no net flow of water from either inside or outside the cell. When the flow of water from and into the cell is in equilibrium, the cell is said to be in a flaccid state. Flaccid cells are found in a wilted plant that has not been watered for a long time. Therefore, it can be said that the surrounding external solution decides the behavior of the cell. Have you ever wondered what happens when we put a twig of white flowers in colored water? The white flowers gradually begin to color. After some time, examine the cut end of the twig. You will spot the colored region through which water was transported inside the twig. This colored region belongs to xylem cells in vascular bundles through which water is transported in plants. Substances often need to be transported across both short and long distances inside plants. Sometimes the site of production of food or absorption of water may be quite far from the plant's storage organs. In such cases, means of transport such as diffusion and cytoplasmic streaming, supplemented by active transport, do not suffice. They can only account for short-distance movements in plants. Moreover, diffusion is a painfully slow process which might take years to transport substances over a distance of 1 meter in plant. Therefore, a special long-distance transport system called a mass or bulk flow system exists in plants to transport water, minerals and food. This system transfers minerals and food at a faster rate than diffusion. As the name suggests, the mass flow system is a process that moves substances in bulk. It arises out of a pressure difference between 2 points. Irrespective of the state substances are in, either in solution or in suspension, they move along at the same pace like in a flowing river. Bulk flow is achieved through either a positive hydrostatic gradient like that caused by a garden hose, or through negative hydrostatic pressure, which can, for example, be caused by suction through a straw. This bulk flow system conducted through the plant's vascular tissues, xylem and phloem, is referred to as translocation. Xylem transports water, minerals and hormones upwards from the plant's roots, while phloem transfers organic or inorganic solutes from the leaves to other parts of the plant. To conclude, the bulk flow system differs from diffusion in 3 ways. First, it transports substances across long distances in plants, unlike diffusion. Second, unlike diffusion, in which substances move independently, the bulk flow system moves all substances at the same pace. Third, diffusion depends on a concentration gradient, while the bulk flow system is achieved through a positive or negative hydrostatic gradient. Therefore, long-distance travel in plants is carried out by a bulk flow system in the plant's vascular bundles consisting of xylem and phloem. When there is a heavy downpour of rain, or when we water our plants in the garden, it is the roots that absorb water for the plant. More specifically, the millions of root hairs present on the root tips absorb both water and minerals from the soil. These root hairs not only increase the surface area for absorption, but also assist in water absorption through diffusion. After water is absorbed, it moves deeper into the root layers by one of 2 pathways, apoplast and symplast. Apoplast is a continuous system of adjacent cell walls in a plant, interrupted only by waxy, suberized Casparian strips in the root endodermis. So apoplastic movement of water occurs through intercellular spaces and permeable cell walls. Water movement is through mass flow and is dependent on the gradient. As water evaporates in the intercellular space, it creates tension in the water flowing inside the apoplast. Mass flow is thus propelled by the adhesive and cohesive properties of water. On the other hand, the symplastic pathway is a system of interconnected protoplasms. In symplastic movement, water travels through the cell cytoplasm, while intercellular movement is through the plasmodesmata. Water movement takes place down the potential gradient. As water enters the cells through the not-so-permeable cell membrane in the symplastic pathway, water movement is slower. However, the symplastic pathway is aided by cytoplasmic streaming, that is, the movement of the cytoplasm. Unlike the rest of the root, the inner boundary of the cortex, that is, the endodermis, is impervious to water due to the presence of Casparian strips. As water molecules are unable to penetrate this layer, they move to wall regions that are not suberized like Casparian strips. Instead, they move inside the other non-suberized cells through the membrane. This water movement too is through the symplast, allowing water to cross a membrane to reach the xylem cells. Therefore, water movement through root layers is symplastic in the endodermis, and it is the only pathway that allows water and solutes to reach the vascular cylinder. Water moves freely again once it is inside the xylem and enters the xylem vessels and tracheids. These xylem elements are non-living and therefore become a part of the apoplast movement, which occurs in the non-living parts of the root. Some plants have additional structures associated with their roots to help in absorption. Mycorrhiza, a symbiotic association between a fungus and a root system, is one such example. Fungal hyphae absorb larger volumes of minerals and water from the soil because they have a large surface area. In turn, roots provide sugar and nitrogen-containing compounds to mycorrhizae. Some plants like pinus seeds cannot even germinate without mycorrhizae. Hence, they are said to be in obligate association with the mycorrhizae. However, irrespective of how water is absorbed, water moves deeper into the root layers through two pathways, that is, apoplast and symplast. When plant roots absorb water and minerals from the soil, they move it in an upward direction to reach different parts of the plant. Have you ever wondered how this upward movement of water and minerals takes place? This is made possible by a combination of root pressure and transpirational pull. Along with the water, various mineral ions from the soil are also pushed into the root vascular tissues by diffusion. As a result, the pressure inside the xylem increases. This positive pressure is known as root pressure. Root pressure can be observed in a plant by a cut stem experiment. In the early morning hours of a humid day, if a soft stem is cut horizontally near its base with a blade, drops of solution ooze from the cut stem. This oozing is caused by positive root pressure. Now try fixing a rubber tube to this cut stem. You will be able to collect and measure the rate of exudation and also determine the composition of exudates. The impact of root pressure is best witnessed during the night and early in the morning when evaporation is slow. At this time, the excess water collects as droplets around the special openings of veins called hydothodes near the tip of grass blades. This water loss in its liquid form is called guttation. However, root pressure itself does not account for the majority of water transport because it can provide only a modest push. Most water movement in tall trees takes place due to transpirational pull. The driving force behind this transpirational pull is transpiration from the leaves. Transpiration is the process of water loss through stomata in the leaves. Unlike guttation, where water is lost in liquid form, water is lost as vapor during transpiration. Transpiration can be witnessed by closing a healthy plant inside a polythene bag. You will notice that droplets of water have formed inside the bag. These water droplets are caused by the loss of water from leaf stomata by transpiration. The water loss due to transpiration pulls the water upwards in a plant stem. Did you know that the transpirational pull generated by this process of transpiration is strong enough to cause water to move upwards even in tall trees by as much as 15 meters per hour? Water transport is also aided by cohesion and surface tension. Cohesion is the mutual attraction between water molecules, and surface tension is the force acting on a water molecule traveling upwards in the xylem. Since cohesion tension helps the transpirational pull, it is also known as the cohesion tension transpirational pull model of water transport. Therefore, we can conclude that together, transpirational pull and root pressure are responsible for the upward movement of water in plants. Did you know that a fully grown tree may lose thousands of liters of water through its leaves on a hot, dry day through transpiration? Transpiration is the loss of water due to evaporation that occurs through pores in leaves called stomata. Stomata are not only the site of water loss for transpiration, they also help in the exchange of oxygen and carbon dioxide in the leaf. They remain open during the day when transpiration takes place and close at night. Their opening and closing is directly impacted by the turgidity of guard cells present near the stomatal aperture. The inner wall of each guard cell is thick and elastic. When turgidity inside two guard cells increases, the outer walls bulge and force the inner walls to take the shape of a crescent. The radial orientation of the cellulose microfibrils in the cell wall of the guard cell helps open the stomata. When guard cells lose turgor, they become flaccid, while the elastic inner cell walls regain their original shape, leading to the closure of the stomata. Transpiration is affected by both external factors like temperature, light, humidity, etc., and plant factors like the number and distribution of stomata. Typically, the lower surface of a dorsiventral dicot leaf has a greater number of stomata. On the other hand, a monocot isobilateral leaf has an equal number of stomata on both surfaces. Transpiration from stomata causes a pull that causes the upward ascent of water in xylem cells. This transpiration pull is made possible by three physical properties of water: cohesion, adhesion, and surface tension. Cohesion is the mutual attraction between water molecules. Adhesion is the property of water to be attracted to polar surfaces like tracheids and vessel elements, also known as tracheary elements, in a xylem cell. Finally, surface tension is the property of water molecules to be more attracted to each other in a liquid state than in a gaseous state. Together, the properties of adhesion, cohesion, and surface tension render water with a high tensile strength. This means that water becomes capable of resisting a pulling force. Also, it gains high capillarity. Capillarity is the ability of water to rise in thin tubes, which is helped by the small diameter of the tracheary elements. This water transported by xylem vessels needs to be moved into the parenchyma cells of leaves, where it is needed for photosynthesis. While water evaporates through stomata during transpiration, a thin film of water appears over the cells, which pulls water molecule by molecule, into the leaf from the xylem. This transport is helped by the water potential gradient. The concentration of water vapor in the atmosphere is lower than that in the substomatal cavity and intercellular spaces. Thus, water diffuses into the surrounding air, creating a pull. Did you know that this pressure generated by transpiration pull is capable of lifting water over 130 meters high in a xylem vessel? Transpiration not only creates this pull for the absorption and transport of water for photosynthesis, it also transports minerals from the soil to all parts of the plant. In addition, it cools leaf surfaces by evaporative cooling and maintains the shape and structure of plants by keeping cells turgid. Photosynthesizing plants require a lot of water. Since water swiftly depletes due to transpiration, the lack of available water may limit photosynthesis. Rainforests are humid largely because of this vast recycling of water from root to leaf to atmosphere and then back to soil. The plant C4 photosynthetic system solves this by increasing the availability of carbon dioxide and decreasing water loss. This system is more efficient than the C3 system, since it not only fixes carbon twice as a C3 system, but also loses half the amount of water lost by the C3 system while fixing the same amount of carbon dioxide. Therefore, transpiration plays an important role in both water transport and photosynthesis in plants. Along with water, plants absorb essential nutrients and minerals from the soil. These minerals are present in the soil as charged particles called ions. Absorption of minerals by plant roots occurs through both passive and active transport. In passive transport, the ions do not require energy and just pass through the cell membrane by osmosis or diffusion. Mineral ions move into the root by diffusion whenever the mineral concentration is higher outside the root. On the other hand, active transport takes place when there is higher mineral concentration inside the roots than outside. Since the ions now move against the concentration gradient, they require energy to cross the cell membrane. Some ions, such as potassium, pass through the cell membrane directly without any energy consumption via either membrane protein pumps or proton pumps. Active transport begins with positively charged ions such as hydrogen being pumped out of a cell. Due to the displacement of these hydrogen ions, negatively charged ions move inside the cell. They enter the cytoplasm of the epidermal cells of the root, using up the ATP molecule for energy. Furthermore, the membranes of root hair cells have some specific proteins such as calcium ATP bases called transport proteins, which actively pump ions into the epidermal cells from the soil. The endodermal cells of the root also have many transport proteins embedded in their plasma membrane. These transport proteins serve as control points, where they adjust the quantity and selectively allow certain types of solutes to cross the membrane to reach the xylem. Also, due to the presence of the suberin layer of the Casparian strip, the root epidermis can transport ions passively in one direction only. The active uptake of ions causes a water potential gradient in roots, which results in the uptake of water by osmosis. After ions reach the xylem either passively or by active absorption, they are transported up the stem to the various sinks inside the plant body by transpirational pull. The sinks inside plants are usually found in its growing regions like apical and lateral meristems, young leaves, developing fruit, flowers and seeds, as well as storage organs. These mineral ions are unloaded at the fine vein endings of leaves through diffusion and are taken up by active transport by associated cells such as mesophyll cells. Plants can also remobilize minerals from older dying parts to young developing parts where they are required. For example, minerals from old dying leaves of deciduous plants are exported to other parts like young leaves before the leaves fall. Phosphorus, sulfur, nitrogen and potassium are readily mobilized as inorganic ions, while some elements such as calcium that are structural components cannot be remobilized. Small amounts of phosphorus and sulfur are also carried as organic compounds. However, most of the nitrogen is carried as organic compounds in the form of amino acids. Moreover, the xylem and phloem exchange small amounts of the materials they carry. Both xylem and phloem elements are responsible for the transport of minerals in plants. Plant food is primarily sucrose. It is transported by a vascular tissue called phloem from a source to a sink. The transport of sugars inside the plant by phloem is called translocation. The leaf is usually the source, since this is where food is synthesized via photosynthesis, while a sink is any part of a plant where food is stored or is required. For example, fruits or roots. However, sink and source roles can be reversed depending on the plant's needs. For example, sugar from the roots may need to be mobilized to a flower bud when they need energy for growth. Therefore, food transport by phloem is bidirectional, both upward and downward. The direction is determined by the concentration of sucrose. This is in contrast to water transport in xylem, which is always upward and unidirectional. A simple experiment called girdling can be used to illustrate the role of phloem cells in trees. In this experiment, a ring of tree bark up to the depth of the phloem layer is carefully removed from the trunk. The portion of the bark above the ring on the stem becomes swollen after a few weeks because the downward movement of food doesn't take place any longer as the phloem cells have been cut. Therefore, it can be concluded that transportation of food takes place via phloem cells. Phloem tissue is made up of sieve tube cells, which are long columns with holes in their end walls. These holes are called sieve plates. Cytoplasmic strands pass through these sieve plates to form continuous filaments. However, most of the cellular functions of sieve tube cells inside the phloem are carried out by companion cells. The fluid that passes through these phloem cells is called phloem sap. it is mainly water and sucrose. In addition, they also transport other sugars, hormones and amino acids from time to time. Phloem sap translocation from source to sink takes place by a mechanism called the pressure flow hypothesis. When plant leaves prepare glucose via photosynthesis, the glucose is then converted into sucrose. Sucrose then moves into the companion cells, after which it is loaded into the sieve tube cells via active transport. This process of loading of sucrose into the phloem causes a hypertonic condition and sets up a water potential gradient. This in turn results in osmosis, which causes water to move into the phloem from the adjacent xylem cells. As osmotic pressure increases in the sieve tube, pressure flow begins, and the phloem sap moves towards the sink, which has low osmotic pressure. This is followed by the sucrose from the phloem sap unloading into the sink cells via active transport. The loss of sucrose produces a high water potential in phloem. This results in decreased osmotic pressure, which causes water to move out of the phloem cells and back into the xylem cells. The sink cells then utilize the sucrose for metabolism or storage purposes by converting it into energy, starch or cellulose. In this manner, phloem cells transport food, primarily sucrose, in plants by the pressure flow hypothesis mechanism.